November 1911, Antarctica. Griffith Taylor stood at the base of a glacier and stared at something impossible. The glacier was bleeding, not rust-colored, not pink from algae or sediment, blood red, vivid crimson pouring from a fissure in ancient ice, staining 200 feet of glacier face, flowing steadily despite air temperature of minus 15 Celsius. Taylor was a geologist, 29 years old, trained at the University of Sydney, methodical by nature. For 3 weeks he'd been leading the western party of Scott's Terra Nova expedition through the McMurdo Dry Valleys, the most Mars-like place on Earth. No precipitation, no humidity, wind-scoured rock, and frozen lakes under pale sun that circled the horizon but never set. They'd been mapping what Taylor would later name Taylor Valley when Debenham spotted it. Called out one word, uncertain, confused. Taylor turned and saw the stain half a mile distant on the glacier face. At first he thought it was shadow, trick of light on blue ice. Then they got closer and the color became unmistakable. Dark red, almost purple in places where it pooled thickest. Flowing. His team stopped 20 feet from the glacier base. Debenham, fellow geologist. Gran, Norwegian ski expert. Ford, petty officer who kept them alive, in conditions that killed the careless. All three staring at the crimson flow like it was a mirage. Taylor approached alone. The ground was unstable, ice hidden under gravel, rocks that shifted, crevasses disguised by snow. He moved carefully until he stood directly beneath the falls. The glacier face rose above him, compressed ice that had fallen as snow before humans existed, and from a crack 40 feet up, liquid poured steadily, spreading down the ice, pooling at the base before soaking into frozen ground. He removed his glove. Stupid in this cold. Exposed skin could freeze in minutes, but he needed to touch it, needed to confirm what his eyes were reporting. His fingers touched the frozen flow. Came away red. He smelled them, metallic, sharp, like blood, or rust, but stronger. He hesitated, then touched his tongue to the residue. Salt exploded across his taste buds. Extreme salinity, bitter metallic aftertaste, and something else underneath. Something organic. Taylor's mind ran through explanations. Iron oxide dissolved in glacial meltwater, simple chemistry. Except, glaciers didn't melt at minus 15 degrees, and the concentration was wrong. This wasn't dilute mineral water. This was thick, vivid, flowing like the glacier had opened a vein. He knelt at the pool, removed a thermometer from his pack and tested the liquid. Minus 2 Celsius, below freezing but still liquid. That explained the flow, but not the source. Where was water coming from inside a frozen glacier? The salinity explained the depressed freezing point. But where was the salt coming from? Antarctic ice was fresh water, compressed snow. There was no salt. Taylor collected a sample in a glass jar. The liquid was viscous, thicker than water. When he held it to the light, it was deep red, opaque, like something had been dissolved in such concentration that the water itself had transformed. He took a second sample from higher on the ice face, scraped frozen flow into another jar, photographed the falls from multiple angles with the expedition's camera, measured flow rate, approximately half a liter per minute, estimated total staining, several hundred cubic meters of red ice. Every measurement raised new questions. The flow was too steady, too concentrated, too red, and there was the smell, faint but persistent, not quite mineral, not quite organic, something in between, something that didn't belong in the most sterile environment on Earth. Taylor made notes in his journal, keeping it inside his coat so ink wouldn't freeze. He sketched the falls, recorded temperature, salinity estimates, flow rate, everything measurable with 1911 technology, but the measurements didn't explain it. They just documented the impossibility. That evening in the tent, wind hammering canvas and temperature dropping toward minus 20, Taylor wrote his theory. Red algae. Extremophile organisms living inside the glacier where pressure melt could exist, producing pigments that colored the ice. He read what he'd written and knew it was wrong. The salt concentration would kill algae, the cold would kill algae, and algae were green or brown, not this vivid crimson, but it was the only biological explanation he could construct. The alternative was purely geological, iron-rich bedrock, oxidized minerals, chemistry, logical, boring, incomplete, because the smell wasn't geological, and the viscosity wasn't geological, and something about the whole phenomenon felt alive in a way Russ never did. Taylor crossed out red algae and wrote it again, left it as his official theory. Wrong was better than no explanation at all. They spent a second day at the falls. Taylor collected samples from different elevations, tested temperature at various points, documented everything he could measure or photograph or describe. On the second evening, he stood alone at the glacier base after the others had returned to camp. The falls flowed steadily, red against white, utterly alien in landscape that was already alien. He realized he was looking at something nobody understood. Maybe nobody could understand with current science, a phenomenon that would require new tools, new concepts, new ways of thinking about what was possible. The thought was unsettling. Taylor believed in geology, believed that observation and measurement and reason could explain anything. But this resisted explanation. Every answer raised more questions. He collected one final sample, sealed it carefully, marked it Blood Falls, a name that was supposed to be temporary but would stick. November 5th. They broke camp. Scott's orders were specific. Complete the survey, return by January. They had 3 more weeks of mapping before they had to turn back. Taylor packed the samples in wooden boxes padded with cloth, wrapped the jars carefully. If they broke, if the glass shattered in the cold, everything would be lost. The team moved out. Taylor looked back once. The falls flowed on, staining the glacier, unchanged by his presence or his questions. The samples would reach England in 1913. Scientists would examine them, find high iron and extreme salt, no algae. Would conclude, iron oxide from subglacial bedrock, geological curiosity, nothing more. The file would close, the mystery would be classified as solved, and for 60 years Blood Falls would flow on in the most remote valley on Earth, running on chemistry nobody understood, fed by water that shouldn't exist, hiding an ecosystem that would rewrite the rules of where life could survive. But none of that mattered in November 1911. What mattered was that Griffith Taylor had found something that made no sense, documented it as carefully as he could, and left it for whoever came next. Behind him, the falls poured crimson against white ice. Patient. Waiting. In 4 months, Scott would freeze to death 11 miles from safety, and the expedition would be remembered for tragedy. Taylor would survive, would become a distinguished geographer, would name features across Antarctica, but he would never return to Blood Falls, never know why it flowed, never learn that his instinct about life was correct, even though his theory was wrong. He would die in 1963 with the mystery unsolved. The answer was already there, flowing steadily in the coldest valley on Earth. It just needed science to catch up with what had been surviving beneath the ice for over 1 million years. The samples arrived in London in September 1913, packed in wooden crates marked Geological Specimens, Taylor Valley. They sat unopened in a museum basement for 9 months. When Dr. Thomas Edgeworth David finally examined them in June 1914, the liquid had frozen solid. Dark red ice in glass jars, whatever viscosity Taylor had noted was gone. The organic smell was gone. What remained was frozen water with visible mineral content. David thawed one sample, ran basic chemical tests, and wrote his conclusion for the Terra Nova Expedition report. Subglacial brine with iron oxide concentration. Coloration due to oxidation upon exposure to air. Geologically interesting but not anomalous. 4 sentences. Mystery solved. David hadn't been there, hadn't seen the flow at minus 15 degrees or smelled that organic undertone, but he was a respected geologist providing a logical explanation. Iron oxide, simple chemistry, case closed. The samples went into storage. Then World War I started, and Antarctica became irrelevant. Taylor read David's conclusion in Australia and felt doubt he couldn't articulate. The explanation was logical, but something about it felt incomplete. He was young, building a career, and questioning your former professor wasn't wise. He published papers on Antarctic geology, became a respected geographer, and rarely mentioned Blood Falls. Meanwhile, the falls flowed on, red against white, year after year, unstudied, unexplained beyond those 4 sentences. Decades passed. The 1920s and 30s brought new Antarctic expeditions to different regions. The dry valleys were too remote, too expensive to reach for science that seemed already done. World War II stopped research entirely. By 1974, when Griffith Taylor died at 93, he'd spent 60 years occasionally wondering if David's explanation was complete. He had no evidence it wasn't, just memory of that smell, that viscosity, that feeling something was alive. His obituary mentioned his Antarctic work. Blood Falls was a footnote. In 1958, geologist Robert Black became the first human to visit the falls. Since Taylor's expedition 47 years earlier, he landed a helicopter during the International Geophysical Year, collected samples with modern equipment, and measured what Taylor couldn't. The results were startling. Iron content 10 times higher than David had estimated. Salinity even more extreme, nearly 40% by mass. Flow continuous at minus 12 Celsius from a glacier that ground-penetrating radar showed was frozen solid hundreds of feet deep. Black's conclusion was careful. Subglacial brine, iron-rich bedrock, oxidation upon exposure, source of brine remains unexplained. That last sentence mattered, because there was no obvious reservoir, No lake. No source for continuous liquid water inside frozen glacier. Half a liter per minute, year after year, coming from nowhere identifiable. Black proposed pressure melt at the glacier base. Ice melting under its own weight, dissolving minerals, emerging hypersaline. Plausible, but the scale was wrong. Pressure melt produced small amounts of water. This was continuous flow requiring a significant source nobody could locate. His paper generated mild interest and no funding. Blood Falls was remote, expensive to reach, and basically explained even if details were unclear. The samples went to laboratories. Geochemists confirmed high iron, extreme salt, deep mineral signatures. They tested for organic material using 1960s methods. Nothing detected. No life. Geology confirmed. Through the 1980s, Antarctica became more accessible. Permanent stations, regular flights, scientists studying climate and ice cores. Researchers visited Taylor Valley, photographed Blood Falls, included it in surveys. Nobody collected new samples. Nobody questioned conclusions from 1914 and 1961. Iron oxide from subglacial brine. Solved. In 1992, graduate student Jill McCookie flew over Taylor Valley on a research flight. She was 24, studying extremophile bacteria, organisms surviving in conditions once thought sterile. She saw Blood Falls for the first time and asked the pilot about the red color. Iron oxide, subglacial brine, been studied since the 1910s. McCookie stared at the vivid crimson staining the glacier. She'd seen iron oxide formations. Rust was orange-brown, dull, weathered. This was blood red, vibrant. Wrong. Has anyone tested it for bacteria? The pilot shrugged. Probably. It's been examined for decades. Back at McMurdo Station, McCookie searched the literature, found Taylor's 1911 report, David's 1914 conclusion, Black's 1961 study, all geology, chemical analysis, mineralogy. Nobody had tested for extremophile bacteria using modern methods, because nobody thought to look. Blood Falls was geology's problem. Life couldn't exist in hypersaline brine at sub-freezing temperature inside a frozen glacier, except McCookie's research was proving life existed where it shouldn't. Bacteria in acid drainage, archaea in Antarctic ice, organisms using impossible chemistry. What if Blood Falls wasn't just geology? She spent 2 years trying to get funding, wrote proposals, applied for grants, got rejected repeatedly. The reviewers' comments were consistent. Blood Falls has been adequately studied. Funding should support novel research, not re-examination of solved mysteries. Solved. That word kept appearing. The mystery was classified as solved despite unanswered questions. Where was the water coming from? Why such extreme salinity? How was flow maintained in frozen glacier? Minor details, not worth money. McCookie kept applying. She reframed the proposal. Not Blood Falls specifically. That wasn't fundable. But a survey of extremophile bacteria in Taylor Valley. Blood Falls would be one of several sites. In 2004, she got approval. Small budget, limited time, enough. 12 years after seeing it from the air, McCookie stood at the glacier base where Taylor had stood 93 years earlier, looked up at red stain flowing steadily at minus 14 Celsius. She collected samples in sterile containers, dark red liquid that would go to Montana laboratories and be analyzed with DNA sequencing technology that could detect organisms in parts per billion, technology that could find life where 1960s methods found nothing. The samples would reveal an ecosystem sealed beneath ice for over 1 million years, bacteria using chemistry older than oxygen, life in conditions that rewrote the rules of possible, but McCookie didn't know that yet. She had samples and questions and determination to challenge 90 years of conclusions. She sealed the containers, marked them carefully. Behind her, Blood Falls flowed, as it had for centuries. The answer Taylor had searched for was in those jars. The answer David had missed because his tools couldn't see it. The answer that would change how scientists thought about life's limits and where to look for it beyond Earth. The mystery was about to stop being solved and start being understood. The drill bit punched through ice at 3 feet per hour. Slow, loud, necessary. McCookie stood beside the rig watching depth readings climb. 10 feet, 15, 20 feet into Taylor Glacier, aiming for whatever fed Blood Falls. April 2004, Antarctic autumn, temperature minus 20 Celsius and dropping toward winter. She had 3 weeks before the last flight out. 3 weeks to drill, sample, and answer a question that had waited 93 years. 6 hours in, at 23 feet depth, the drill hit liquid. McCookie signaled stop. At this depth, this temperature, there shouldn't be liquid. Ice should be frozen solid. But the pressure gauge showed resistance changing. The bit was in water. She disconnected the drill assembly with hands that shook slightly inside heavy gloves. Not from cold, from recognition that she was about to touch what Taylor had touched, what David had dismissed, what Black had measured but not understood. The sampling tube went down sterile, came up dark red. She held the collection jar to light. The liquid was almost purple, thick, viscous, exactly what Taylor had described. That evening at McMurdo Station, in a lab the size of a closet, Makuki prepared her first slide. Her hands were steady now. This was procedure, dilute sample, place under microscope. Observe. She adjusted focus and saw movement, not Brownian motion, not crystals forming, movement, deliberate, organic. The field of view filled with bacteria, hundreds of them, rod-shaped, flagellated, swimming through brine that was supposed to be sterile. Makuki sat back from the microscope, stared at the wall, looked again, still there, still moving, living bacteria in hypersaline brine, at sub-freezing temperature from inside a frozen glacier. She prepared another slide from a different sample. Same result, and another, same. Life where every textbook said life couldn't exist. Makuki ran basic tests with shaking hands. Cell count, millions per milliliter. Motility, active. Metabolism markers, positive. These weren't dormant spores waiting for better conditions. They were thriving. She sat in the tiny lab listening to the building creak and wind and tried to process what this meant. If bacteria could survive here, extreme cold, extreme salt, complete darkness, zero oxygen, then they could survive anywhere. Europa's subsurface ocean, Enceladus's ice sheets, Mars permafrost. Every environment scientists had dismissed as too hostile might need re-examining, but first she needed to understand how. What chemistry sustained life in conditions that should kill it? Over the next 2 weeks, Makuki collected samples from different depths and locations, built a profile of what lived beneath the glacier. Every sample contained bacteria, not sparse survival, abundance, an entire ecosystem sealed under ice. The chemistry was strange, extremely high iron content in two different oxidation states, reduced and oxidized iron in the same sample, and sulfate, high concentrations of sulfur compounds. She recognized the pattern from deep ocean vents, chemosynthesis, bacteria pulling electrons from iron, using them to power metabolism, releasing oxidized iron as waste. Other bacteria reducing sulfate, generating energy from chemistry instead of sunlight. But ocean vents had heat, volcanic activity, energy gradients. Blood falls had none of that, just cold darkness and brine, yet somehow minimal energy from iron and sulfur chemistry was supporting abundant life. Makuki ran calculations late into the night. The energy yield was barely enough for basic metabolism, but the system was closed, nutrients recycled, nothing wasted, an ecosystem running on chemistry as old as life itself, the kind of chemistry that might have sustained Earth's earliest organisms, before photosynthesis, before oxygen, before sunlight became biology's primary energy source. Blood falls wasn't just supporting life, it was supporting ancient life. Using metabolism that hadn't changed in billions of years. But how long had this been isolated? She looked at glaciology data. Taylor Glacier ice above Blood Falls was old, very old. Radiocarbon dating suggested the ecosystem had been sealed for at least 1.5 million years. 1.5 million years. An ecosystem in darkness, recycling the same elements, surviving on minimal energy, since before modern humans existed. The bacteria had been there when early hominids first stood upright in Africa, through ice ages and warm periods, and the entire span of human evolution. When Taylor first saw red liquid pouring from white ice and tried to understand what he was looking at, generation after generation, adapting, optimizing, surviving. Makuki sent samples to laboratories in Montana and California, places with DNA sequencing equipment that could identify species and analyze genomes. The results came back over following months. Diverse bacterial community, multiple species in symbiotic relationships. Genetic analysis showed they were related to known bacteria, but had diverged significantly. A million years of isolated evolution had created unique adaptations, cold shock proteins, salt tolerance mechanisms, iron processing enzymes optimized for darkness. The discovery made headlines in 2009. Ancient ecosystem beneath Antarctic ice. Life survives million years in darkness. Astrobiologists took immediate notice. Blood falls proved complex ecosystems didn't require photosynthesis. Chemistry alone could sustain abundant life if the system was stable and closed. It changed the search for extraterrestrial life, expanded the definition of habitable. Anywhere with liquid water and chemistry might support organisms, even in eternal darkness, even sealed beneath ice for geological time. But one question remained. Where was the water coming from? Makuki returned to Antarctica in 2006 with ground-penetrating radar, mapped the subsurface beneath Taylor Glacier, and found it. A lake, Subglacial lake approximately 4 kilometers long, buried beneath hundreds of feet of ice. Liquid water maintained by pressure, geothermal heat, and extreme salinity preventing freeze. Blood Falls was overflow, the place where pressure forced brine up through cracks in the glacier, a window into an ecosystem existing in complete darkness below. The lake had been sealed for millennia, possibly millions of years, supporting life that had adapted to conditions Earth hadn't seen since the Archean eon. It was a time capsule, a demonstration that life didn't need what scientists had assumed essential. Standing at the glacier base where Taylor had stood a century earlier, McCookie understood what he'd felt, that instinct something was alive. He'd lacked tools to prove it and framework to explain it, but he'd been right. The red stain wasn't rust. It was metabolic waste from bacteria oxidizing iron. The flow wasn't just meltwater, it was overflow from an ancient reservoir. The organic smell Taylor had noted, bacterial biofilm, the viscosity, microbial material suspended in brine. Blood Falls wasn't bleeding, it was breathing. Had been breathing for over 1 million years. Would continue long after humans were gone. Taylor had documented a mystery in 1911 with the tools he had. David had explained it in 1914 with the understanding available. Black had questioned it in 1961 with better technology. And McCookie had solved it in 2004 because science had finally developed methods sophisticated enough to see what had been there all along. Not geology, not rust, life. Ancient, resilient, thriving in darkness beneath the ice of the coldest valley on Earth. February 2010. Jet Propulsion Laboratory, Pasadena, California. Dr. Chris McKay stood before a room full of NASA engineers and astrobiologists, showing them a photograph of Blood Falls. "This," he said, pointing to the red stain on white ice, "is why we're going to Europa." For 15 years, the Europa mission had been proposed, designed, funded, defunded, and shelved repeatedly. Too expensive, too risky, too speculative. The arguments against sending a probe to Jupiter's moon were always the same. Even if Europa had a subsurface ocean beneath miles of ice, it would be dark, cold, isolated, sterile. McKay advanced to the next slide. Microscope image of bacteria from Blood Falls. Rod-shaped cells moving through hypersaline brine. "Life," he said, "thriving in conditions identical to Europa's predicted subsurface environment." The room went quiet. These were people who'd spent careers designing missions to find extraterrestrial life. They understood what they were seeing, not speculation, not theory, proof that life could exist in exactly the conditions Europa offered. One of the engineers raised her hand. "How long has that ecosystem been isolated?" "Over 1.5 million years. Completely sealed. No sunlight, no oxygen, just chemistry." The engineer nodded slowly. McKay could see her recalculating, mission parameters changing in her mind. Europa wasn't a long shot anymore. It was following evidence. 3 months later, NASA announced renewed funding for the Europa Clipper mission. Launch target 2024. Objective: determine if Jupiter's moon could support life. The press release mentioned Blood Falls specifically, cited McCookie's research, called the Antarctic discovery a template for life in darkness, but the implications went far beyond one mission. In astrobiology departments across the world, researchers were rewriting assumptions about habitable zones. For decades, the search for life had focused on planets at the right distance from their stars. Not too hot, not too cold, liquid water on the surface. Earth sat in that sweet spot. Mars was borderline. Everything else in the solar system was too far, too frozen, too hostile. Blood Falls changed the equation completely. Surface conditions didn't matter. Life could exist sealed beneath ice, powered by chemistry, independent of starlight. The habitable zone wasn't a thin band around stars. It was anywhere liquid water and chemical energy coincided. Enceladus, with its geysers suggesting subsurface ocean. Titan with hydrocarbon lakes. Even Pluto might harbor liquid water kept unfrozen by radioactive decay in its core. The universe had billions of frozen moons and planets, places dismissed as sterile because they were too far from stars, too cold, too dark. Every one of them was now potentially habitable. Dr. Kevin Hand, astrobiologist at JPL, put it simply in a 2011 interview. "Blood Falls taught us that we've been looking in the wrong places. Life doesn't need sunlight, it needs liquid water and chemistry. That's everywhere." The search shifted. Mars missions stopped focusing on surface rocks and started planning deep drilling, looking for sealed aquifers where bacteria might survive using ancient chemistry, exactly like Blood Falls. But McKay understood something the press releases didn't capture. Blood Falls wasn't just about finding life elsewhere. It was about understanding what life required at its most fundamental level. He'd spent 40 years studying extremophiles, bacteria and acid mine drainage, archaea and boiling springs, organisms in Antarctic ice. Each discovery had pushed the boundaries of possible, but Blood Falls was different. Blood Falls bacteria didn't just survive extreme conditions. They thrived, built complex ecosystems, maintained diversity, all on chemistry so minimal it barely met the thermodynamic requirements for metabolism. They represented what life looked like at its most efficient, most resilient, most independent of external energy. They were probably similar to Earth's earliest life, before photosynthesis evolved, before oxygen existed, before biology became dependent on starlight, living on iron and sulfur and chemistry as old as the planet. Blood Falls was a window into Earth's past. 4 billion years ago, before cyanobacteria started producing oxygen, this was how everything lived. In darkness, using chemistry, independent of the sun. That window mattered because it showed what life could revert to, what it could become again. On frozen moons or deep underground or anywhere sunlight couldn't reach. The universe was mostly dark. Stars were rare, planets bathed in friendly light were rarer, but chemistry was universal. Water was common, ice was everywhere. Life might not be rare, just hidden, sealed beneath surfaces. Living in darkness on chemistry humans were only beginning to understand. In Antarctica, Makuki continued her research. She returned to Blood Falls every austral summer, drilling new samples, monitoring how the ecosystem changed, or didn't change, over years. Other scientists joined her. The subglacial lake became one of Earth's most studied extreme environments. Each expedition revealed new complexity, more bacterial species, more symbiotic relationships, more evidence that minimal energy could support surprising diversity if the system was stable. In 2014, Makuki led an expedition that placed sensors in the lake, temperature monitors, chemical samplers, equipment that would record conditions continuously for years. Standing at the drill site, watching the sensors descend into darkness, she thought about Taylor, how he'd stood at this glacier 93 years earlier, collected samples with glass jars, and tried to understand what he was seeing with 1911 science. He documented the mystery as carefully as he could, left it for whoever came next, died without knowing the answer, but convinced something was alive. She was standing on his shoulders, using tools he couldn't have imagined. Answering questions he'd asked when asking them seemed pointless because the technology to investigate didn't exist. That progression mattered, because there were other mysteries waiting, phenomena dismissed as geology or physics, that might be biology in forms humans hadn't recognized yet. Science advanced by people who looked at solved problems and asked if the solution was complete, who questioned comfortable conclusions, who kept investigating even when funding was denied, and reviewers said the mystery was adequately studied. Blood Falls had been adequately studied in 1914. Case closed, just rust. Except it wasn't rust. It was life. And it took 90 years of advancement, technological and conceptual, before humans could see what had been there all along. That lesson was broader than astrobiology. It was about humility, about recognizing that current understanding was incomplete, that the universe was stranger than scientists assumed, that impossible was often just improbable until tools improved. The bacteria in Blood Falls subglacial lake didn't care about any of this. They'd been there before humans evolved, would be there after humans were gone. They didn't care about NASA missions or scientific papers or implications for the search for extraterrestrial life. They just lived, generation after generation, using iron and sulfur, sealed in darkness, breathing in ways that redefined what breathing meant. And on the glacier surface, Blood Falls flowed on, red against white, visible sign of invisible life. The window Taylor had found and Makuki had opened, proof that life found ways to exist where science said it couldn't, that the universe was full of possibilities humans were only beginning to see, and that the coldest, darkest, most isolated places might be exactly where to look for answers about what life could become when pushed to its absolute limits. The falls flowed, ancient, patient, waiting for the next question. The next generation of scientists with better tools and new frameworks, still bleeding, still breathing, still teaching anyone who looked carefully enough that impossible was just another word for not yet understood.